So the question is, how much of the microbial world do we know about? Well, we know a lot, right? Think of all the antibiotics we produce. Think of all the fermented and brewed products that we make. Think of all the things we make with microbes. Think of all the microbes that are important in disease and geochemical processes and so forth. We know a lot about those. There are about 6,000 named and characterized species of bacteria. I told you already that in lab we get about 1 in a million. I think even that's an overestimate. And so one of the issues is, okay, so how many species of insect are there? I don't even know the answer to that, but it's in the millions. 506 million. Very good. So what if each one of those species of insects has only a single specific bacterial symbiont? Organism that grows on its cuticle or in its gut or something like that. We know that this is the case for mammals. Each one of you has each species of mammal has a distinct species of Staphylococcus that grows on their skin, Staph epidermidis in the case of Staph epidermidis strain hominis in the case of humans. And you can draw a phylogenetic tree of staph and a phylogenetic tree of mammals, and the two are superimposable. They co-evolve together. That's what we're doing. The same is certainly true of insects. What if each one of those only has one specific symbiont? That means there's 3 orders of magnitudes of bacteria right there, just in that one example, than the ones we know about. 5 billion instead of 5,000. Think of the entire world. I mean, there must be phenomenal numbers of bacterial species. Think about environments that we haven't even touched yet. Deep aquifers, cave systems, hydrothermal fields, clouds, the surfaces of organisms. I mean, there are all kinds of environments that we don't know anything at all about, and all of those have microbes that live in them. The biggest problem with counting bacterial species is defining what a species actually is. So, when you talk about plants and animals, a species is a breeding population, right? But bacteria don't breed. In fact, most creatures don't breed. Bacteria don't breed, archaea don't breed, most protists don't breed, at least not in the traditional sense. It's only in plants and animals and a few other creatures that you have this tight linkage between reproduction and the sexual exchange of DNA. In most protists, actually most organisms generally, do exchange DNA. They do, in a population of some kind, mix genes up. There's a strong selective pressure for this. Because as a population, your ability to be resilient depends on your ability to resort genes so that you can pick the ones that work best under the conditions that you're in. But it's really only in a small number of organisms that you force that in every generation. So what is a species in the microbial world? We don't know. There's no definition for that. One way to think about it is by hybridizing DNA together to see how similar they are. And traditionally, anything where you have DNA-DNA hybridization of 70% or more is considered to be the same species. Don't want to apply that to plants and animals because all animals are the same species by this definition. Probably that's not a good choice, right? In terms of ribosomal RNA, oftentimes you see the term operational taxonomic unit, OTU, and most commonly this represents 97% identity of ribosomal sequences. This is used because, particularly early on in the process of high throughput sequencing, anything where you're only getting 100 nucleotides at a time, you might see 3 differences that are sequencing errors or that sort of thing with a fair frequency. And so that was kind of the limit of resolution of the technology. But again, all mammals are within 3% identity in terms of sequence similarity of ribosomal RNA. And I doubt that you and ferrets would be considered the same species by any useful definition. So what? I mean, one way to do it is to look to see how easily two different clones of a microbe will exchange DNA by homologous recombination. That's a pretty good definition. It turns out that there's no easy boundary for that, because whether they exchange that DNA easily or not depends on what regions of the DNA you're looking to exchange. So, this issue of a species concept in bacteria and other unicellular creatures is a really tough one. And it's not one that people have looked at very hard, and I think that's a mistake. The entire notion of natural selection comes from an examination of just exactly what is a species in plants and animals. That's where Darwin got his notions of evolution, was looking at a species and saying, wait a minute, these groups aren't really entirely distinct units the way we think of them. The boundary between species is mushy. We saw this in plant hybridization. We saw this in things like ring species, where population across a geographic area were different enough that in different parts of that area, they were considered different species. You saw this as interbreeding between related things that people used to think of as species, red fox and gray fox, for example. And in variation between individuals that are considered the same species. It's really remarkable how much variation there is within most species. We don't think about this very much because humans are almost an exception to this. Humans are remarkably homogeneous in terms of their morphological and genetic diversity. We're not a very diverse species. Most species have a lot more morphological and genetic diversity within the population. And Darwin saw this looking at barnacles and looking at the same species of barnacles, and every individual has different morphological idiosyncrasies. In the doctor's office, if you go into the hospital and you've got some non-specific problem, almost the last thing they'll do is shove you through an MRI machine and look blindly for what might be wrong. One of the reasons for this, of course, is it's really expensive. But the real reason is because they know they'll find something. They know they're going to find 2 or 3 idiosyncrasies in your anatomy. And those probably have nothing to do with your illness. All of us have idiosyncrasies in our anatomy and physiology. And that's perfectly normal. How many of you have done a lot of dissections? Yeah, so you see stuff like that. I remember when I was in high school, we dissected a dogfish for some class I took. And in dogfish, the pancreas is this elongated organ that runs right along the small intestine. And it's big at one end and small at the other. Well, mine had 2 pancreases. The 2 lobes were completely separate. And they were big at both ends, and there was nothing in the middle. There was no connection between the 2. Perfectly normal. You're the same way. And so this issue of what a species is not as clearly defined, even in the macroscopic world, as you think it is. Species are not these silos that are completely independent of each other. Think about this in terms of time. So when a species, over time, splits off like so, at this time point, you can say you've got 2 species. There's no interbreeding between them. At this point, it's 1 species. But what about here? If these are 2 species, is this organism the same as that one? Same species or a different species? Those boundaries are arbitrary. Those are things that humans decide, make those decisions about what they put in their group. And this goes back to one of the first lectures I gave you. That species is a taxonomic unit. It is an artificial collection of organisms based on a person's notion of putting things together. It's just like putting together brass nails and steel nails and brass screws and steel screws, and that's okay. These are taxonomies. These are artificial ways to organize things. Species are no different. But there ought to be, the functional definition of a species in the macroscopic world does have to do with these breeding populations. And as arbitrary as they are, it's still a very useful concept. One that we lack entirely in the microbial world. And I think that's something that we really ought to be looking at harder. Because we can't count species. If nothing else, we can't really measure diversity in these populations without having some way of categorizing them and looking at them. There are some other ways that are really interesting based on trees, and we'll talk about those later on. All right, so we're done for the day.